So for those that are joining for the first time, welcome to the third live event of the Translational Neuroimaging Educational Program. And this is a program featuring a series of talks meant to engage the neuroimaging community and brain science community to enhance the translational application of computational neuroimaging techniques in promoting human brain health. So the program is nested within the Enigma Lifespan Working Group, and it hosts expert lectures, training workshops to promote the use of neuroimaging techniques to advance medical and scientific knowledge, all centered around brain health. And this is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The program provides opportunities to expand skills, initiate collaborations, and facilitate networking in a welcoming learning environment. So this is, again, the core team that has organized these events, and the core advisory committee as well, who has helped kind of shape the program. And today we'll actually be hearing a talk from soon-to-be postdoctoral fellow Lukas Röll, who is joining us from the LMU Munich and works together with Dr. Daniel Kaiser at the Ludwig-Maximilians-University. I'll provide a more detailed description of kind of his background and what he does and what he will be talking about today at the end of this introduction. So just in case you've missed any of our past events, you can tune into them via our YouTube channel, which is available here. It's just youtube.com and then at translational-neuro. And all of the events that we host, we plan to put them online for others to access and refer back to for future reference, and hopefully initiate collaborations amongst multiple different researchers at different levels. It's great to see that there has been so much interest from around the world for this initiative. We're really excited to see where this journey takes us. If you'd like to receive updates on for future events and upcoming events, and any news that we post, you can sign up for the mailing list. The website is just translational-neuro.org. And then we're also on Twitter or X, as well as LinkedIn, and provide updates there as well. And then just as a preview for the next event that we have planned after this, which will take place on May 14th at 6 p.m. Pacific time, that would be 9 p.m. Eastern time. There's usually a conversion that's available on the website if you RSVP to find out what time this event would take place based on your time zone. You can RSVP for the event using the QR code, or again, you can go to translational-neuro.org to sign up for the event. And the next session will be hosted by Dr. Chao Yan from the Institute of Psychology at the Chinese Academy of Sciences. And he will present the DIRECT Consortium and REST Meta MBD project, which has the aims towards imaging-guided precision treatment of major depression. So we look forward to seeing all of you attend the next event as well. And stay tuned for more exciting events to come. If you have any methods or work that you would like to present within this platform, of course, you can reach out to Dr. Guy or myself in order to discuss potential ideas to share with other researchers through this platform. Okay, so today's session is titled How to Measure Functional Connectivity in the Brain, a comprehensive empirical exploration of connectivity metrics. And it will be presented by Lukas Röll, who works together with Dr. Daniel Kaiser, who heads the Neuroimaging Core Unit at the Ludwig-Maximilians-University in Munich. and is also a part of the advisory committee, as I mentioned. Lukas is currently transitioning to a postdoctoral fellowship in the psychosis working group of the Department of Psychiatry and Psychotherapy of the Ludwig-Maximilians-University in Munich. He recently submitted his PhD dissertation titled Effects of Aerobic Exercise on Functional and Structural Brain Patterns in Schizophrenia, and will be defending his thesis in April. So good luck for that. His research focus involves investigating neural, genetic, physical, and environmental mechanisms that explain clinical improvements in schizophrenia using both interventional and multimodal longitudinal neuroimaging studies. There's a ton of information to cover, as you can see the outline here, and we will leave open about 10 minutes at the end for Q&A and discussion. So yeah, I'd say let's go ahead and get started. Thank you.
I'm gonna have you repeat that. What was it?
Thank you, Shalaila, for this kind introduction. That's very nice. It's a big honor to get the opportunity to give this talk today and presenting our work. Now you see my slides, I assume, right? Yeah, great. Okay. Yeah, as Shalaila said, normally I'm not such a methodological researcher, but besides my main PhD topic, I came across this concept of functional connectivity since we were using it or calculating it. And then I was wondering about this concept and how one could evaluate that. And this kind of led me to this side project that I did together with Daniel Kaiser, besides my PhD. So the title of today's presentation, as you can see, is how to measure functional connectivity using resting state fMRI. And the whole project is basically an empirical exploration of different connectivity metrics. So the central concept of this talk is functional connectivity. And as I'm not quite sure how familiar the audience is with functional connectivity or resting state fMRI research in general, I would briefly give an overview on the basic concept of functional connectivity. So it is defined as the statistical interdependence of the BOLD signal, the blood oxygenation level dependent signal of two or more brain regions or voxels or vertices. The BOLD signal in turn is said to be an indirect measure of neural activity. There's lots of debates on that, but for this talk, we just assume that this is true. The assumption which could be derived from that is that, and when we come back to functional connectivity, is that the higher the similarity of neural activity of two or more brain regions is, the stronger these brain regions are assumed to be functionally connected. So this is basically the assumption behind the concept of functional connectivity. And to make it a little more precise, I will talk about how to measure similarity and the different strategies to do so. But first, just to visualize what I try to present here in theoretical bullet points, it's like that. You have your functional brain image, your AP sequence, and then you extract the BOLD signal from either voxels or vertices or regions. And then you get here these tiny time series. Then I use the pointer. You get this time series for each ROI, which could be a voxel, a vertex, or a region. And this time series is just telling you or provides you information about the activity over time that a specific region shows. This is what you do in the end after all the pre-processing stuff, all the denoising stuff. In the end, you extract the BOLD time series as a proxy of neural activity during the scanning period. And what you do then is you quantify the similarity of these BOLD time series, meaning you try to compute, or you try to examine how similar the neural activity of this ROI is in comparison to this ROI and to this and to this and so on. And in the end, you end up with a connectivity matrix just telling you how similar the BOLD time series of two regions, voxels, or vertices are. And the most common way to quantify similarity is using either Pearson's correlation or partial correlation. There are lots of, there are more sophisticated strategies to deal with this kind of data. However, the vast majority of studies just use Pearson correlation or partial correlation. And this is exactly, when I first read that during my PhD, I was kind of wondering that the brain is so beautiful and so complex. There are so many synapses, so many interactions that are going on, and we are just, I don't know, you're just correlating two very indirect measures of neural activity. So I was wondering, isn't there more sophisticated stuff? Isn't there alternative approaches how you can do that? And may these approaches work better than just correlating these bold time series? And then I came across this paper here, Unifying Pairwise Interactions in Complex Dynamics, published last year in Nature Computational Science. That's a group of physicists, I think from Australia, and they in their paper provide a very comprehensive overview of all the possible opportunities to quantify similarity between different types of time series. And when I say different types, I really mean it is an interdisciplinary work, meaning that they're not only focusing on neuroimaging time series, but also on time series from economics, etc. So that's a very comprehensive and very great work. And in the end, they suggest 237, I think, different metrics to quantify similarity between different kinds of time series. They have a preprint available on Archive, which was already published in 2022. That's a kind of fun story because when I started this project I'm presenting today, I was at first trying to code and to write a Python code in order to calculate all these metrics that I wanted to investigate by myself. And then after, I don't know, 2 months of work or so, I stumbled over this preprint, and then I was saying, okay, shit, there other people already did that. And the best thing about that, they already provide a Python toolbox, a Python module called PySpi. I don't know how to pronounce it. Here you see the links for that, and this is basically a toolbox enabling you to calculate any type of similarity metrics for your time series that you want. And it's very, very comprehensive. It's very, very complex. The only thing that you need in advance is to extract the bold time series from your data. And then in the end, you can use this toolbox to calculate different types of similarity metrics. And this is exactly what we did. And we, in the end, came up with 2 different research questions. The first research question was, when we apply typical research approaches that are typically applied in functional neuroimaging, for instance, such as brain behavior associations, meaning that you correlate a functional connectivity pattern that you observe to a certain behavior, such as cognitive performance. This is very typically done in neuroimaging or functional neuroimaging. And the second thing is case control comparisons, meaning that you try to identify disconnectivity patterns in the brain and kind of in search for so-called biomarkers, for instance, for psychiatric disorders that can explain or that are kind of inherent to a specific disorder like schizophrenia. And these are two very, very typical research approaches in functional neuroimaging. And we were just wondering, how do the results and the resulting conclusions vary depending on the FC metric that you choose? That's a very simple research question, I think. And the second part, because I mean, it might be kind of boring to show that that results may vary. We wanted to examine if you have a case where there's really apparent neural decline in the brain, for instance, induced by aging or induced by malignant tumors, then which metric captures this apparent neural decline most accurately? So this was the second part of our project, of our research question. And as you can see, the FC metric is, again, a central concept, a central methodological thing to consider in our work. So which metrics did we choose? I already mentioned that this toolbox enables you to calculate 237 different metrics. We didn't take all of them. We selected 20 representative metrics that have already been used in functional neuroimaging in the past. And those 20 metrics basically came from or are from 4 different categories. First thing is correlational measures, like I already mentioned, the Pearson correlation, partial correlation, Spearman correlation, Candle's tau, and cross correlation. These were the 5 correlational measures that we tested. When you want to get into the details of how these measures are computed, I would recommend to go into the original paper by Cliff and colleagues, or also in our preprint we described them very briefly. The second category that we were focusing on were distance metrics: Euclidean distance, city block distance, cosine distance, dynamic time warping with the so-called Itakura constraint, and dynamic time warping with the Sakaue-Chiba band. Again, if you want to get into the mathematical details on that, I recommend either to read our preprint or to read the original paper. There, these measures are well described. And then the third category was frequency measures. So we focused on magnitude coherence or coherence magnitude, phase coherence, the phase locking value, the phase slope index, a directional measure, and also a directional measure, so-called spectral Granger causality. And the last category that we considered was information theory measures, meaning mutual information, 3 different types of mutual information, which basically measures the information flow, linear and nonlinear, between 2 time series, and then transfer entropy with the Gaussian estimator, and another type of transfer entropy with a so-called Kraskov-Stögel-Bauer estimator. The transfer entropy with a Gaussian estimator is basically the normal Granger causality. Okay, so these were the 20 metrics that we examined, but on which databases? So we used 4 data sets in total. First, the Human Connectome aging data set, consisting of in total 725, but I put the numbers here that were included in the final statistics. Then the MBB, the so-called Mind, Brain, and Body data set from the Max Planck Institute in Leipzig. Then our own cohort that we collected over the past 3 or 4 years, the so-called clinical deep phenotyping cohort, consisting of healthy participants, but also patients with schizophrenia. And then again from Open Neuro, the brain tumor connectomics data set, where we basically analyzed the data of a few patients that had a grade 3 tumor. So these were the 4 data sets. All the analysis that you will see in the upcoming minutes are based on. And for the first research question that I mentioned, we only used the CDP cohort from our clinic. So here, just a brief reminder, first research question: if you have a typical research approach in functional neuroimaging, such as brain behavior associations or case control comparison, how do the results vary depending on the functional connectivity metric of choice? What we did here is we used the CDP data set, we used the T1 and T2 weighted images and the resting state fMRI images, pre-processed them with fMRI prep, and then did the denoising with NiLearn, extracted the ball time series, and then we calculated functional connectivity between hippocampal and frontal seeds, because we wanted to, for the brain behavior association, we were focusing on functional connectivity, on the association between functional connectivity on the one hand and cognitive performance on the other hand. And so we choose this hippocampal frontal connectivity because it has been shown to be associated with several core cognitive functions in for every in pathological samples, but also in healthy controls. And then we calculated multiple linear Bayesian regressions, or Bayesian multiple linear regressions, the order is like that, and using cognition, calculated via the bugs, which is just a cognitive battery, and then hippocampal frontal functional connectivity as one predictor, and age and sex as covariates. We used only the healthy controls from the CDP dataset for that. And for the other part, so for the case control comparison, we did basically the same pre and post-processing strategy, and then extracted the bold time series from the 7 Yeo networks, calculated functional connectivity based on 20 different connectivity metrics, and again calculated Bayesian regressions where we used group, so schizophrenia versus controls, age and sex as predictors, and the between network connectivity. So for instance, functional connectivity between the default mode network and the salience network, etc., calculated for 20 different connectivity metrics as the dependent variable. Exactly, this was the first part. For the second part, we used these 3 data sets. Again, just a brief reminder, the second research question: which functional connectivity metric captures apparent neural decline most accurately? First factor that we identified was aging, and for that we used the HCP data and the mind, brain, and body data set. And first, we wanted to ensure that there is age-dependent neural decline in our cohorts, in our 2 cohorts. So what we did is we used the structural data, processed it with FreeSurfer, extracted the volumes of several hub regions in the brain. These were based on a previous paper from graph theory saying that specific regions in the brain are particularly connected to all other regions, and they call them hub regions. So we selected the volumes of those, just averaged the volumes of all the hub regions, so that we had one score in the end, providing you the average volume across all hub regions. And then in the end, we again calculated a Bayesian multiple linear regression with volume in the hubs, age and sex as dependent variable, age and sex as predictor. Then we computed another Bayesian linear regression with cognition as dependent variable, the volume in the hubs as predictor, and sex as one covariate. We did not include age here because we wanted to see which metric in the end, which connectivity metric in the end is most sensitive to age-related cognitive decline. Yeah. We did exactly the same for the DTI data. Here we used MRTrix to process all the data, build the structural connectome, compute for between all hub regions, computed the mean structural connectivity across all hub regions, and basically had the same statistical model in the end, a very simple statistical model that like for the volumes, but only with structural connectivity between hubs as dependent variable here and here as predictor. Then we used for the HCP data, we used also the ASL data. We processed them with Oxford ASL, extracted the perfusion values based on the Desikan-Kiliani atlas from FreeSurfer, the perfusion values from the hub regions, again calculated mean perfusion, and then did basically the same statistics like here, but only with perfusion as the neural outcome of interest. For the MBB data set, they had no ASL data available, so here for ASL we only have the HCP data. And then for both data sets, we basically did exactly the same approach for the functional data. In the end, calculating mean functional connectivity between hub regions, but this time for each functional connectivity metric that we examined, and then had the same Bayesian regressions that we calculated. So this was very briefly the main part, I would say, of the analysis. Then the second part Here, we used the brain tumor connectomics data set. They had DTI and structural MRI, structural and functional MRI data available. We processed the data again with MRTrix, computed the structural connectivity matrices. But this time, we did it like this, that we used all the regions that were close to the tumor of these patients. And all the tumors were located in one hemisphere. So in the end, we calculated a hemisphere-specific measure of structural connectivity between regions that were very close to the tumor, and then exactly the same. We took exactly the same regions, but in the other hemisphere where the tumor was not located, and then made just a simple descriptive comparison between structural connectivity of those regions in the left hemisphere versus right hemisphere. And we basically did exactly the same for the functional data, but again, for 20 different metrics. So we did a lot. As I mentioned, and as this is an educational session, I would like to talk about Bayesian statistics very briefly, just a few slides, because as I said, especially for this aging stuff, we computed Bayesian linear regressions. And I don't know how many of you are familiar with Bayesian statistics, so I would give a brief introduction into that, the upcoming, I would say, 5 to 10 minutes, and then I will come back to the main project onto our findings. So Bayesian statistics. You will notice I'm a big fan of Bayesian statistics. I think the best approach is to start with an example. So when we talk, for instance, about breast cancer, we see that the prevalence of breast cancer is approximately 1% in the population. When you go to a breast cancer screening, they will use a certain instrument to detect if you have breast cancer or not. And this instrument is quite good. So it has a sensitivity of 90% and a specificity of 91%. So the main question is now, you go to the doctor, you get a positive test result. What is the probability that you really have breast cancer? And when you calculate that, imagine you have 1,000 patients, 1% prevalence, meaning 10% cases, 10 cases in your sample of 1,000 patients. 990 people or patients do not have anything. Yeah, do not have breast cancer. Now you go to the doctor and you see, okay, the instrument that is used has a sensitivity of 90%. So for those 10 who really had breast cancer, there will be 9 positive tests in total and 1 negative test. And for those 990 cases without breast cancer and a sensitivity of 91% of your instrument, you will have 89 positive tests and 901 negative tests. And then imagine now the situation is you went to the doctor, you got a positive test result, and most of the doctors that are asked or are asked about this scenario will say, okay, if the test result is positive, we would assume the probability that you really have breast cancer is around 80, 70, 80%, but it's not. It's 9.2% in total. You have 9 positive tests, true positives here, and you have 89 positive tests of people which do not have breast cancer. So in total, when you calculate the probability, it's 9.2%. This is very low, right? And now I come to Bayesian statistics, because the cool thing about Bayesian statistics is you consider the prior knowledge that is available for your particular research question in the actual statistical test that you are performing. Yeah, we could think of the screening like a statistical test, a case control comparison or something. Yeah, and when you do that in Bayesian statistics, you can consider the prevalence or the size of the expected true effect in your single statistical test. And this is, I think, a great thing. And to illustrate that a little better, what you do in Bayesian statistics is you define the so-called prior. So meaning, I don't know, you're doing a case control comparison, for instance, on functional connectivity between patients and controls. And then you have some rough idea about how the true effect should look like. So what you can do in Bayesian statistics, you define a prior that enhances the probability of certain values that you expect, here the effect size, a priori. And then you do your experiment, you observe a specific effect with a confidence interval, and then the likelihood is estimated based on the data that you provided. And from both, from the likelihood and the prior, in the end, you get the posterior, which is then basically the effect that you expect, or the final effect that you expect to be true. Yeah, and the posterior for the next researcher who's going to answer a similar research question, for the posterior will be his or her new prior. So this is the basic idea of Bayesian statistics. I don't go into the maths here, just the principles. And the main outcome that you get in the end from Bayesian statistics, and now it gets important also to understand our later results, is the so-called Bayes factor, which is kind of the equivalent of the p-value in frequentist statistics. And the Bayes factor is basically the probability odds between the probability to observe your data, here indicated with D, under the assumption of the alternative hypothesis, divided by the probability to observe your data under the assumption of the null hypothesis. So it kind of compares the probability to observe your data, assuming that the null hypothesis, the alternative hypothesis is true, to the probability that to observe your data, assuming that the null hypothesis is true. Sorry. But to make an example, if you observe in the end a Bayes factor of 7, for instance, this would just mean it's 7 times more likely to observe your data under the alternative hypothesis in comparison to observe your data under the null hypothesis. So it's a continuous measure of evidence. There were some authors who proposed a categorical scheme for that, saying that if you observe a Bayes factor of 1 to 3, it is called anecdotal evidence. Then 3 to 10, moderate evidence. I mean, this always depends if you want to do something like this, to categorize such a continuous measure, but it is possible, and it's, I think, reasonable to have a common language on Bayesian statistics. So just very briefly in comparison, if you compare Bayesian, the statistics here have the Bayes factor who fights against the p-value from frequentist statistics, and then I might be a little bit biased here, but the clear advantage is in Bayesian statistics you can consider prior knowledge, which you can't in frequentist statistics for your particular test. You can quantify evidence for the alternative hypothesis or for the null hypothesis, whereas in frequentist statistics you can only provide evidence against your null hypothesis. Here the keyword is the falsification principle proposed by Popper. Then you have a continuous measure of evidence. The p-value is also continuous, but it is kind of damned to be discrete. You all know this significance threshold of 0.05. A very great thing about Bayesian statistics, there's no real multiple testing situation that you have, because you don't, in the end, you don't make a binary test decision. So you just say, okay, for my particular research question, we could provide, let's say, anecdotal evidence. And then people will say, okay, this is only anecdotal, and it's kind of, yeah, the evidence is not very good. We need maybe a replication or so. But you don't do anything like, okay, now the p-value is below 0.0, or the Bayes factor is above 3, and now we have an effect. This is not true in Bayesian statistics. And then another thing, maybe a quick side note, the interpretation of the confidence interval is much easier. You can say that the true value, given an appropriate prior, you can say that the true value really falls within with a probability of, let's say, 90% within your confidence interval, which you can't for frequentist statistics. There, the interpretation is much more complicated. If you want to know more about Bayesian statistics, there are here numerous papers. Just take a screenshot. Mostly psychologists which are using that. I think our psychology chair at the LMU, this year they start teaching Bayesian statistics in the first semester and doing frequentist statistics only as a side lecture or something. If you want to get a practical introduction to Bayesian statistics, there's a very nice blog from Felix Schönbrot, translated to Nice Bread, who's a professor at our psychology department, where he offers lots of opportunities to experience the Bayes factors and explains everything in a very good and easy way. There's another tutorials available here at this homepage. Then Shiny apps, which can kind of teach you to p-hack, but also how to manipulate Bayes factors, etc. So it helps you to get a better impression of how how Bayes factors behave. And then when you want to do Bayesian statistics in your particular research, it's relatively simple. The BRMS package in R is available. It's with regard to the syntax, it's very, very similar to the classical approaches. But it's easily applicable, I think. So now we go back to the main research question after this brief talk about Bayesian statistics. As you may remember, the first research question was, we have typical research approaches in functional imaging, for instance, brain behavior association or case control comparisons, and how do the results vary depending on the functional connectivity metric of choice. So as I said, we tested 20 functional connectivity metrics. Here are the correlational metrics, the distance metrics, frequency metrics, and the information theory metrics. And what you basically see here is the beta coefficient of the particular functional connection that we were investigating. Remember that it was hippocampal frontal connectivity. And I extracted the beta coefficient from the Bayesian regressions here, and we have the Bayes factor on the y-axis plotted. And what you see, for instance, when you use Pearson correlation, you would detect strong evidence or a Bayes factor of 10 for the association between the left parahippocampal gyrus and the left caudal anterior cingulate gyrus. And so you would find something like, okay, this association here is very strongly or clearly associated with cognitive performance. Yeah. The bad thing is when you use partial correlation, there isn't anything. You wouldn't find that. You would find it again with Spearman correlation, with Candle's tau. You would not find it when you use cross correlation. And you see, one could go through all these findings, but in general, we see, okay, there is variation between or in other words, the choice of connectivity metric matters. And the same is for case control comparison. If we have if you have a positive beta here, it means that that there's a hyperconnectivity between these networks in patients with schizophrenia compared to controls. And again, you would see, if you use partial correlation, you would observe, okay, patients seem to have a hyperconnectivity between the visual network and the dorsal attention network, only with at a moderate evidence strength, but you would not observe it with Pearson correlation. And again, we could go through all these findings here. But I would say it's the same conclusion. The choice of metric matters. So they vary a lot. It's not very surprising because they are mathematically completely different. And the big question is, what is the ground truth? Meaning, which finding can we trust on these findings? And we tried somehow to answer it. even though I get to that at the later stage, it is difficult. So the second research question, as you remember, in case of a strong neural decline induced by aging, which metric captures this neural decline most accurately? So what you see here is first the structural analysis, the mean volume in Hubs, and you have the age plotted here based on the HCP cohort, and you see a very, very strong effect, base factor way above 100, clear, the confidence interval clearly in the negative range, meaning the older the people, the lower the volume. This is what we, it's a very, very clear effect, and what we would expect to observe. We also see this association for mean structural connectivity measured by DTI, but the effect this time is much smaller. You see it here, the beta is about minus 0.18. Here it is minus 0.6. And you see the regression line is much flatter. We did the same for the ASL data and also observed a relatively strong effect, meaning age-related decline in mean perfusion in Hubs. And now getting back to our research question, which metric does capture this association most accurately? And indeed, we have Pearson correlation, which works quite well, partial correlation as well, but the base factor is here only at 2.1, which is supposed to be anecdotal evidence and might not even statistically significant if you have done it in a frequentist approach. Spearman correlation, candle tau work quite well, as well as cross correlation. You observe exactly the same pattern. For the distance metrics, except dynamic time warping with an Icahn constraint, you observe that as well. The other metrics here, apart from mutual information with a kernel density estimation, are not working or are not capturing this association or this neural decline. We tried to replicate it in the MBB data set from Leipzig. Here we didn't have ASL, but we also had the volume. This time we had to divide the groups in old and young because the real ages of these people were not published. They only published the age bins. This is the reason why you see box plots here. But you see the same volume, lower mean volume in the hub in the old people compared to the young people, and lower structural connectivity, although the effect is again a little smaller in old people compared to young people. And then when you look at the functional connectivity metrics, in this case, what you observe is that again, the correlational measures perform quite well, but again, partial correlation, not as good as the other ones. Again, a base factor of only 2.8. And the best metrics for this data set is cross correlation with a base factor way above 100, and in this time dynamic time warping with the Icahn constraint, which didn't show a signal or this association before in the HCP data. For the frequency metrics, we seek something completely going in the opposite direction in this data set. And then last slide regarding the aging. Now you see the cognitive composite score for the HCP data from the NIH toolbox on the y-axis, and you see the volumes, mean volumes on the x-axis. And what you see is a positive association, meaning that the higher the volume is, the better the cognitive performance. Keep in mind that we did not control for age here because we wanted to mimic the age-related cognitive decline. We don't see any association when we look at structural connectivity between hubs. That's interesting. So structural connectivity in at least using our analysis, we do not find an association with cognition. And for perfusion, and that's quite interesting, we find the strongest effect. Meaning when you measure brain perfusion with PC-ASL, yeah, then you observe here a beta of 0.19, a base factor way above 100, and this effect, this association with cognition is even stronger than for the volumes. And for the functional connectivity stuff, you see again Pearson correlation, but again only a base factor of 1, which is very weak. Yeah, this pattern. So there's not really one metric capturing this association. When we use the strategy that we used, how to calculate everything, yeah, that we have to keep in mind that. Exactly. So this was regarding the aging. Now we're going to an individual patient level, looking at these malignant tumors. I briefly guide you through the slide here. You see here the tumor, in this case in the right hemisphere, yeah, in the right temporal and insular cortex, superior temporal mainly. And yeah, you see the tumor here. In the tractography, you see this gap. Yeah, so a clear gap in the tractography. And here you see the regions from the brain atom atlas that we chose to compute structural connectivity between regions close to the tumor on the ipsilateral hemisphere in comparison to the contralateral hemisphere. And this is just single patient data. And what we saw, what we also see here, obviously, and you can also quantify it. So mean structural connectivity between in the right hemisphere where the tumor is located is reduced. In the right hemisphere compared to the same regions in the left hemisphere. When you when you apply that for controls, for 10 controls, an average across all these 10 controls that were provided in the data set, you don't see this difference. And then for this patient, you see, okay, that's again the correlational measures and the distance measures, apart from dynamic time warping with an Itakura constraint, work quite well. And in this case, also the phase slope index. And when you go to the next patient, now it gets a little weird. This is the tumor is here in the parietal cortex. You see the gap here. You see the regions that we selected here. And then again, when you compare that right hemisphere where the tumor is located to the left hemisphere where the tumor is not located, you see this clear difference in this patient, which is also mimicked by, yeah, or can be seen here and here. And in healthy controls, it's kind of flipped, but I would say this is a coincidence, and it's rather a little more balanced, I would say. And for this particular patient, we see that the correlational metrics behave completely opposite. So like in the distance metrics as well. And the only metrics which behave the same like the structural data is transfer entropy with a Gaussian estimator and mutual information with a kernel density estimation. So this is kind of weird because patient 4 before, it worked quite well for the patient before, and for this patient not. Or maybe this could reflect something else, maybe a kind of compensation process or something in connectivity on a functional level that only this patient here showed. And the last patient that we looked at, again, same play. You see the tumor here, you see the gap, and then you see the white matter difference, not in controls. And here, basically, nothing is showing up for the correlational measures. You could imagine that this is a difference and this, which kind of is similar to that, but not as huge as, or not as large as for the first patient, for instance. Okay, so what should we all derive from, or what should we derive then from, or how could we summarize all these findings? I would say generally, we across data sets, especially when looking at the aging effects, we see that there is a superiority of correlational and distance metrics in capturing neural decline, which is kind of good because, yeah, especially correlational measures are mostly used in our research, functional neuroimaging research. Interesting is that partial correlation in our case was a little worse. This is kind of contradicting to a simulation study done in 2011 by Smith and colleagues, where they said partial correlation is better. But I think there might be a good explanation because we use this summarizing functional connectivity scores across regions. And when you use partial correlation, I think or when you use Pearson correlation, you give more weight to those regions that have many, many connections. And those regions might also be more affected by aging, for instance. This could be an explanation, potentially. Then what we observed is the role of the scanning sequence. We saw that, for instance, dynamic time warping with an Itakura constraint worked well for the MBB data set, but not in the HCP. So there might be something depending on the scanning sequence. This has already been shown in simulation studies on functional imaging data, where they also showed that the length of the scanning period that you use and also the repetition time, etc., plays a substantial role when computing functional connectivity. One could argue, based on our tumor data, that also the regions may play a role. So, and the type of connections that you are interested in, because we had this one patient where, for instance, transfer entropy mimicked this decline in white matter, this white matter decline better. And could be that, I don't know, that in the regions that we were investigating for this patient, based on his or her tumor location, that those regions, that there are more directional measures or directional connections within this network, and this leading to the transfer entropy to be superior because it's a directional measure. And we saw this very weird variation between individuals, which we cannot really explain at the moment. And as a side finding, we observed that brain perfusion measured via PC-ASL seems to represent a relatively robust neural and representation of neural and cognitive decline, which is kind of exciting because it's not commonly used so far in research. We now implemented PC-ASL in all of our studies in clinical neuroimaging. What are the limitations? I think there are numerous limitations of what we did. I think one could still argue, did we really capture the ground truth? So we tried because we looked for age-related neural decline in all the other modalities, such as volumes, structural connectivity, perfusion, and then we somehow assumed that this should also manifest in impairments in functional connectivity. This is the assumption the whole project is based on, but it's not necessarily true. As we saw, maybe for this one patient showing a compensation process also. And functional connectivity and structural connectivity are not the same. Yeah? So they are related, but it's not the same. And the problem is a little, when we started this project, we hoped in the end to find one metric which is kind of superior in comparison to all the other metrics, and then to give a clear advice when to use which metric, or which metrics to use. We cannot really do that based on our data because it's very heterogeneous. We only focused on 20 representative metrics. One could extend our approach going for all the 237 metrics that are proposed by Cliff and colleagues. We didn't include any network modeling approaches. And we computed linear regression, Bayesian linear regression. And also Dr. G and Shalaila, you showed in your papers or with the centile brain that age-related decline is not necessarily linear, or it is for sure not. So we could also think of applying different models. However, we wanted to reduce the complexity, and we still saw that there is that the effect is still observable also with linear models. Okay, I'm coming to the end, but now I think it's one of the most important points I want to make this evening in Germany. There's a theoretical functional connectivity framework proposed by Reid and colleagues in 2019, published in Nature Neuroscience. And they are differentiating between three properties. The first property is the so-called theoretical property. So this is everything, all the properties about the neural system you want to investigate. Meaning, for instance, is it a neural system where you have directional connections included? Is it monosynaptic or polysynaptic, etc.? So all the characteristics of the neural system that you examine. this is something you have to, we should all, I think all people doing functional connectivity research should be aware for our particular project, what kind of system are we investigating? The second thing they're saying is that the methodological property, meaning you have to define a little more precise, or we all have to define a little more precisely why we choose certain methods, such as the functional connectivity metric or the temporal and spatial resolution of our data, to make the best inferences about the theoretical property. And lastly, there's the confounding property, meaning that we should define for every experiment that we are doing the sources of confounds that are potentially relevant to investigate the theoretical property. I think our empirical data somehow confirms that, because just a brief example, we used here as a methodological approach, as one approach, we used partial correlation in order to control for unmeasured neural sources. Yeah, this is basically the idea of using partial correlation. But then we observed that for the theoretical property that we chose, meaning this averaged connectivity between different systems, this approach, this choice of methods, and considering this here was not really good, yes, in the end. So I would say our empirical data somehow confirms the importance of this theoretical framework. So how to conclude? Honestly, yeah, the brain is really beautiful, and it is still beautiful and complex. We are correlating. There is much more sophisticated stuff we could think of doing, but it's not necessarily better. I would conclude with that, and to make it a little more seriously, I think it's very important for future functional connectivity studies to embed their particular approaches in the theoretical framework that I just presented. And I think as a side finding, PCASL may be a promising MRI modality to study brain perfusion in clinical neuroimaging. This is something we should definitely consider. We are doing that now at our clinic. And here I'm at the end. I want to thank Daniel Kaiser, one of my MRI supervisor, then the shared first author, Stefan Wunderlich, who cannot make it today, I think. Then my supervisor for my PhD project, Dr. Peter Falkay. Then I want to thank Professor Shi, because he proved all the stats that we did and all the mathematics. And then I want to thank the psychosis group at the Department of Psychiatry at the LMU Hospital in Munich. Here again, if you want to go for the details, here's the barcode or the QR code for the preprint that we published. Okay, I hope that it wasn't too long.
Okay, yeah, so thank you so much.